I wanted to give you a great biology example of those gated channels. So here's our phospholipid bilayer again, with our little fatty acid tails here. And this will be the outside of the cell, and this is the inside of the cell. And here is a channel protein. Now, some channels have gates on them, and the gates are closed. And the opening of the gate requires a certain chemical, and we call this chemical a ligand, L-I-G-A-N-D. So that is a specific chemical that will cause a gate to open. This channel is a sodium gated channel. So this is the kind of thing that you would find in a nerve cell. If you hit your hand and you think, wow, that hurts, the reason why is it's receiving a signal, and the signal is going to your brain. So we got to talk about nerves for a minute. Nerves extend out. If I just draw a neuron, neurons tend to look like this. I know you think I'm such a fantastic artist. Here is the initial area where a signal could be picked up. Each of these are called dendrites. And dendrites have on them receptors. So a receptor, we talked about, receives a certain molecule. That molecule it receives is what we call a ligand, a specific chemical that can cause a change and cause a reaction to occur. What it's actually picking up, the specific ligand in a nerve, is a neurotransmitter. So I'm going to draw a little green thing here, and I'll have several of them in here. This region is called the synapse. If you remember, there's always a space in between one nerve cell and the next nerve cell. So these neurotransmitters are present in the synapse, and they can be picked up by receptors. The neurotransmitters are the ligand. They are the chemical message. They are what's going to be picked up by the receptors, and they're actually going to allow that sodium-gated channel to open. A normal nerve cell, this is what we call the axon of the, this is what we call the cell body. And this long part right here is what we call the axon of the cell. And the axon is where that change has to occur to actually pass a message down. That message is going to be electrical, and electrical really just means a difference in charge. The normal setting is to have a lot of sodium, Na plus ions, outside of a cell, and inside of a cell there are potassium ions. But there's not as much positive charges within the cell as there are outside of the cell. One of the other reasons why DNA is negative, and that is also within this cell. So this cell normally has a negative 70 millivolt charge. It's polarized, which means there's a difference in charge from the inside to the outside. Now, if I'm going to pass a message down this, and it's going to be an electrical message, I need to change that. I need to depolarize it, meaning get rid of the difference in charge, and I got to let some of these NAs in. So I want to go back to this picture. The ligand is my neurotransmitter. So I'm going to put NT for neurotransmitter. And when a neurotransmitter binds right here with the gate, it opens it up and sodium come rushing in. Now, is this active or passive transport? Have you seen an ATP anywhere attached? That's your first clue. It's definitely passive. The other reason why it's passive, there is a high concentration of sodium outside of the cell and a low concentration inside. So this movement of sodium is just a great way to show how a sodium-gated channel works and to describe to you what a ligand is, a chemical messenger such as a neurotransmitter that will open a gate. Open the gate and allow this to come in. And we're going to do a lot more with nerves this year, so you'll have a better understanding of what I mean about polarization and how this changes and what changes after that. But I wanted to give you something that was pretty specific in terms of a channel protein opening.